Okay, so this is chapter 3, lecture for Anatomy and Physiology 1. This whole chapter is about cells and the cell cycle, the cell parts. The purpose of this slide is okay, that's better. The purpose of this slide is to just show all the different cell types. So, for example, we can have these adipocytes, which are really large cells, or nerve cells look like this, or some nerve cells look like this one. Muscle cells can be really long, like up to 18 inches. And this is a smooth muscle cell, and this is a skeletal muscle cell. So this is more just to show that how those muscle cells look and how they're measured. So these are micrometers, the unit of measurement. Composite cell. Composite cells aren't really cells. There's no cells that look like that. Okay, so the composite cells just show all the possible organelles that you would find. So this is a composite animal cell. So this is showing the flagellum and the nucleus, Golgi apparatus, cilia. So this is all the different things that could be on an animal cell. There are no animal cells that have all of these. Okay. Plasma membranes. Plasma membranes is the outer layer and of the cell. So it is what holds everything in. It's selectively permeable because the plasma membrane allows some stuff to enter the cell, but not others. And same thing, some stuff to enter or exit. So that's the selectively permeable part. It also receives signals from the body, and maybe the cell's receiving a signal to make a particular hormone or to make a particular fluid, like mammary glands will receive a signal to make milk. And also it can send signals out to the body. Plasma membrane is mainly made up of lipids. So remember from the last unit, those phospholipids and proteins, transmembrane proteins, and there are some carbohydrates on there. Okay. Plasma membrane is a phospholipid bilayer. So it has, and we looked at these phospholipids in the last unit. And the phospholipid had a polar head, that hydrophilic head, and those nonpolar tails. Okay, so there's the head, and then there's the nonpolar hydrophobic tails. And remember that the phospholipid bilayer means there's two layers of these phospholipids. Things that can get through the phospholipid bilayer can just pass through are things that are soluble or lipid soluble. Things that are water soluble can't pass through because this is a hydrophobic area in here. Cholesterol is there to stabilize the membrane. It keeps it fluid in cases of when the membranes heat up or cool down. The plasma membrane gets a little, this just touches the surface of it, but the plasma membrane, the things in there in the plasma membrane can move, like the phospholipids can move within the plasma membrane, and those transmembrane proteins can move within the plasma membrane. And there's a whole group of functions of those membrane proteins. Okay, and we'll talk about a few of those. Carbohydrates are found on the glycoproteins, and those can act as a cell recognition, almost like the carbohydrate chains act like little flags on the cell membranes to tell the body that, hey, this cell should be here. Okay, so a white blood cell won't come and attack it, hopefully if it has the right carbohydrate chains on there. So let's look at a few of those. So here's the phospholipid bilayer. Okay, that's that phospholipid bilayer right here, where you have the heads towards the inside and the heads towards the outside, wherever the hydrophilic environments are, those watery environments. Transmembrane proteins are called transmembrane proteins because they're made of proteins and they go across the cell membrane. And these ones have these are glycoproteins because they have a carbohydrate chain on them. Okay, and then there's the, it's kind of hard to see, it's probably easier to see in your books, all these cholesterol molecules helping to maintain the fluidity of that cell. All right, now we're going to talk about the specific organelles within cells. So the first one is the cytoplasm. The cytoplasm isn't just a watery environment or a gel-like environment, as you may remember from biology, but it's the cytoplasm has the cytosol, which is the fluid portion, but also has the organelles. Okay, and those organelles have very specific functions within the cell, that do something for the cell. So they all have their own little jobs. Cytoplasm contains a cytoskeleton, and that cytoskeleton is made up of proteins and microtubules cytoskeleton helps to direct the flow of traffic through the cell and then support the interior of the cell. Ribosomes function in protein synthesis. So ribosomes are where proteins are made. Ribosomes have two parts. They have two subunits. There's subunit A and B. Those subunits are made of protein, and the other one's made of RNA. And remember from previous unit, RNA is ribonucleic acid. Okay, so ribosomes function in protein synthesis. The ER, there's actually two parts to the ER. First of all, the ER itself is attached to the nucleus. And the rough ER is called rough because it has all these ribosomes studded on it. Let's change my pen color. So the rough ER has all these ribosomes studded on it, and that's what makes it appear rough. And since that's where the ribosomes are, this is the site of protein synthesis. The smooth ER doesn't have ribosomes, so that's why it's called smooth. And the smooth ER is where lipid synthesis takes place, but it's also where drugs and poisons are detoxified. Okay, so smooth ER is where drugs and poisons are detoxified and where lipids are synthesized. Rough ER is where proteins are synthesized, because that's where the ribosomes are. Vesicles are just membranous sacs, usually made up of a phospholipid bilayer. And vesicles are used to store and transport substances throughout the cell. So when a protein is made in the rough ER, it gets transported in a vesicle. The Golgi apparatus looks a lot like the ER, but it's not attached to the nucleus. So that's how you can tell the difference there. And the Golgi apparatus is where proteins are modified and packaged for export from the cell. Okay, so when a protein is made on the rough ER, the protein's carried to the Golgi apparatus, in a vesicle, and the Golgi apparatus will modify the protein, add things to it, fold it a certain way, and then package it for export from the cell. Mitochondria. Mitochondria have a double membrane. So they have an inner membrane and an outer membrane. And the mitochondria is where cellular respiration takes place. Cellular respiration is how we make our ATP. And ATP, if you remember, is that energy for the cell. It's what the cell uses for energy. So mitochondria is where that happens. So this is where cellular respiration takes place. Lysosomes. Lysosomes are vesicles, basically, and they have digestive enzymes in them. And the lysosomes, digestive enzymes, can be used to break down bacteria that's brought into like a white blood cell, any worn out organelles, anything in the cell that is no longer needed, or cellular waste. So think of Lysol, that's like that spray stuff that's used to clean, Lysol and lysosomes. Peroxisomes have specific enzymes in it. And peroxisomes, for example, are used to break down like hydrogen peroxide. And hydrogen peroxide breaks down hydrogen peroxide is a metabolic waste product and is broken down by a specific enzyme called catalase. So catalase is this enzyme that breaks down hydrogen peroxide into water and oxygen. Okay? And peroxisome is what carries that catalase. Centrosomes are organelles that are made up of two centrioles. Centrosomes are only found in animal cells during mitosis. So when animal cells are undergoing cellular division, they will, the centrosomes will show up. Cilia. Cilia are extensions of the cell membrane. They have microtubules that move the cilia. Cilia are like flagella, but they're shorter and there's more of them. One of the functions of cilia is to move stuff over the cell. So, for example, in your respiratory system, the cilia line the respiratory tubules to move mucus through your respiratory tract. And then in the reproductive system, the cilia move the egg from the ovaries into the fallopian tube down into the uterus. Flagella, usually there's only one flagella if a cell has a flagella. Sperm cells for humans are the most common example. Flagella also have those microtubules in their tail. Here's a sperm cell, and the flagella is the tail. And there's microtubules in there that beat the back and forth, this whip-like fashion to move the sperm cell. Okay, the nucleus. Nucleus is a place where we store our genetic material. It is nucleus is surrounded by a nuclear envelope. Nuclear envelope is just a double membrane. It's almost like its own cell membrane. Nucleus has these nuclear pores through it, and the nuclear pores is how stuff enters and leaves the nucleus. Inside the nucleus, we have this thing called the nucleolus. Nucleolus is where ribosomes are made. So RNA and protein are found in here, so we can make ribosomes. And the chromatin is found within the nucleus, and the chromatin is just your DNA with proteins called histones, and those histones help to wind up the proteins. And the chromatin is just there for to carry the genetic information.